Good morning, everyone. I am Valentina Tonelotto, an employee of a Spanish company, Xenopat, who have been seconded to University College Dublin, where I joined the laboratory of Professor Brendan Kennedy. My secondment started in October 2021, and it will last 12 months. I am a biologist from Italy, and I obtained my PhD in the University of Padova, where I led a project to study the roles of collagen-6 using zebrafish animal model. After this academic experience, I worked as a biologist in a clinical laboratory, and I then had the opportunity to join Xenopat as a lab and market researcher. From there, I have been seconded in UCD, and in Professor Kennedy's group, I am working to find therapeutic targets and biomarkers to improve patient outcomes in uveal melanoma, which is a poor prognosis cancer of the eye, which frequently spreads to the liver, with up to 50% of the patients developing metastatic uveal melanoma. So there is a urgent need for effective treatment and detection of this devastating disease. My main expertise are related to cell biology, molecular biology, biochemistry, and in vivo experimentation, and my research interests are related to translational medicine. Indeed, my aim is to find effective treatments for rare and poor prognosis diseases. So the measure aims of my secondment are to evaluate whether ex-vivo uveal melanoma liver metastases exhibit a patient-specific therapeutic response to CysLT receptor antagonists. So we will collect liver samples from patients under surveillance for metastatic UM and from OPDX mouse models. So models for liver uveal melanoma generated using tissues from metastatic patients. We will grow these explants, these tissues as explants, and we will treat them with CysLT receptor antagonists to evaluate how these drugs affect cancer secretome. Moreover, I will verify whether extracellular vesicles can be biomarkers of uveal melanoma, mediators of uveal melanoma cancer hallmarks, or therapeutic targets through CysLT receptor antagonism. Till now, I mainly focused my attention on this second aim, and so in the last months, I learned how to isolate EVs from plasma using the sucrose cushion ultracentrifugation protocol that I summarized here in this scheme. Of course, I also performed some analysis to evaluate whether I was effectively able to isolate EVs, and by Western blot, I detected an enrichment of specific markers associated to EVs, while by electron microscopy, I was able to detect these specific particles. I mainly worked on this second aim also because, unfortunately, COVID-19 emergency caused a delay in collecting uveal melanoma liver metastases. But in the meantime, I had the opportunity to be involved in additional projects, among which the testing of new CysLT receptor antagonists and the evaluation of the effects of CysLT receptor antagonists on specific intracellular signaling pathways. Overall, all the parts involved in this secondment will benefit from it. Indeed, Xenopat will benefit from the new techniques that I will learn to grow the liver samples from OPDX mice, while the group of Professor Kennedy will gain a unique collaboration with a Spanish company which successfully generated 4 rodent models for metastatic UM. Concerning my professional and personal development, I am having the opportunity to work on a new relevant clinical field, so I'm learning new techniques, and I will also have the chance to attend national and international meetings. I am also building a new interaction with academic and non-academic experts, with patients and with society. So also society will benefit from this secondment, as I will have the opportunity to contribute in building a uveal melanoma community whose aim is to find effective treatments for this devastating disease. Thank you very much.